Hello. All right, welcome back. Hope you had a good weekend. So this is our last lecture before our first exam. So our short honeymoon year is almost over. I know it's quick to have a first exam. I talked about the reasons that I wanted to do that. I wanted to have a first exam that was worth a little bit less. It's going to be a little shorter than our other exams. So hopefully it will be for the best. I am going to have the exam covering a lot of what we're lecturing on today. So I'll let you know when the cutoff is. And I won't normally lecture kind of right up until an exam. I know that's only giving you a couple of days to study today's lecture, but it just worked out that way this time, and it won't likely again. Normally you will have more like 4 or 5 days at least to study all the material. So I do want to make sure I get through that material today. So we'll get going here. The one other thing I wanted to say was that I am concerned that there's going to be a good number of you that are, positive or testing positive or sick or in quarantine for the exam on Wednesday. So I think I am going to offer that by respondus if you have one of, an excused reason. If you can't come to campus, you're showing symptoms. Because the idea of trying to get like 15 of you in for a makeup, I've tried to do that before and it just about drove me crazy. So those of you, that are listening to this recording, or if any of you start having symptoms, just be in touch with me and I will make a respondus version available. I really, I want you to only do the respondus if you absolutely can't be here. So I think it's better if you are here. I know there are problems with respondus, technical problems, other problems. If you would rather, if you are quarantined, say till Thursday or Friday, I would rather have you come in and do a makeup maybe later. So I can certainly arrange that as well, but let's use respondus as an option as well. So again, hopefully you will all be here. We will be crowded in here. There's 120 of us, so we will be kind of crowded. Try to put your stuff off to the side so it's kind of not in your way. And again, just the first 15 minutes of lecture on Wednesday, we'll do that exam, and then I'm going to lecture a little bit after that. Something else that I don't normally do. But okay, so questions. Don't forget your homework is due Wednesday as well, up through this chapter 3 material. It's due at the time of the exam. So okay. And I still have office hours tomorrow afternoon as well. So, okay. So last time we finished up taxonomy, we talked about the contributions of Linnaeus and then Woese and Fox in terms of, sequencing ribosomal RNA to make up this new level of taxa, the domains. And then we started into our structures of prokaryotic cells. So we're going to continue there. We got kind of partway through talking about the cell wall, which is a structure that we spend more time on probably than any other structure. We talked about how all bacteria have this compound called peptidoglycan. Okay, we don't very often say always or never in biology, but if a bacteria has a cell wall, it will always have some peptidoglycan in it. And if an organism is not a bacteria, it will not have peptidoglycan. So that's kind of a hard and fast rule. We labeled the gram-positive cell wall, what it looks like. So remember, lots of peptidoglycan. Really, the only other things that are unique to the gram-positive cell wall is that teichoic and lipoteichoic acid. The gram-negative cell wall then is a little bit more complicated. All right, so remember, this is going to have a thin layer of peptidoglycan. Okay, only about 10% of the cell by dry weight. Sorry, about 10% of the cell wall. All right, and that number for gram positives was somewhere between 40 and 80%. So thicker peptidoglycan certainly for the gram positives. And then the other thing that's really unique to the gram negative cell wall is that it's going to have an outer membrane. Okay, so I'll show you a graphic of this in a minute, but it's got the cell membrane, which we haven't really talked about yet, and then that layer of peptidoglycan, and then outside of that is this outer membrane. Okay, and so that outer membrane, it's a phospholipid membrane, phospholipid bilayer, kind of similar to cell membrane. Within that are porin proteins, and those are just necessary to transport things through that membrane. So all sorts of things in order so they can get into the cell. And then a compound called lipopolysaccharide. And we often abbreviate that and refer to it as LPS. Okay, again, this is unique to the gram negative cell walls. What is lipopolysaccharide? Well, there's several different polysaccharides associated with it, as well as a lipid A. And sometimes when we have a structure that's really important, we find that we have several different names for it, which makes it a little bit more confusing. But lipid A is also known as endotoxin. And that in itself is confusing. When we first discovered endotoxin, we thought it was something that bacteria cells were making inside their cell. So that's why it was given the name endotoxin. Well, this is actually a structure, a compound that is on the outside of the cell. It's actually lipid A is endotoxin. And that is important because when the cell wall breaks down, okay, so when then if this is a bacteria cell that's inside some host, that lipid A can have some really detrimental effects to the host. Okay, it can cause an inflammatory response. Okay, so the lipid A is something that gram-positive bacteria don't have. So right off, there's a difference that we have to, in terms of treating a bacterial infection. It's kind of first thing we ask, is this a gram-positive infection or a gram-negative infection? We would use different drugs probably to treat a gram-negative infection. And we would have to take into question, whether this lipid A, whether this endotoxin is going to cause a problem. What I forgot to say is when is the cell wall broken down? A little bit when the cell divides, but mostly when the cell dies. Okay, when that bacteria cell dies, the cell wall starts to break apart, and that's when the lipid A gets released and can cause this inflammatory response in the host. What do I mean by inflammatory response? Things like fever, some inappropriate blood clotting, just kind of lots of other inflammatory effects. We're going to talk more about inflammation more towards at the end of the semester. Okay, so let me show you a graphic of the cell wall. So once again, let's try to label the one from the book. It's not perfect, that's why there's another graphic that I want to show you as well. But here, first thing we come to here, this is the cell membrane. We're going to talk more about that in a minute, kind of briefly, really. And then we've got this small layer of peptidoglycan, right, much smaller than the gram-positive. And then this whole outside of that's referred to as the outer membrane. Okay, this structure here that kind of looks like a couple of paper towel rolls, maybe that's that porin protein that I referred to. Okay, they need to have, it's just a special transport channel to get things in and out of the cell. And then these kind of hair-like structures here, that's the lipopolysaccharide, okay, in the very, very outside of the cell. Okay, I like this other picture better from a different textbook, so I thought I would show this as well. Of course, it draws out the layers a little bit differently. Cell membrane or plasma membrane here, a layer of peptidoglycan. And then this outer membrane, I really like, especially up here, how it draws out the lipopolysaccharide. Okay, they've got two polysaccharides. That's the kind of hair-like structure sticking out. And then the lipid A is actually embedded there in that outer membrane. Okay, so not causing any problem at all when that cell membrane's intact, or sorry, when that cell wall is intact. When the cell divides, so we talked a little bit about some of the things that has to happen when the peptidoglycan is going to, you know, get a little bit bigger. When that happens, that outer membrane is going to maybe be disrupted just a little bit, but when the cell dies, that cell outer membrane, as well as the rest of the cell wall, is going to break apart, and that's when that lipid A is released. Again, another name for that lipid A is endotoxin. Okay. All right. So basically, that's the basic parts of the gram-positive and gram-negative cell wall. There is actually a third type of cell wall that's much less common. Most bacteria cells are either going to be gram-positive or gram-negative. Okay, there's always an exception, so we have to talk about the acid-fast cell wall. This is a kind of cell wall that's unique to really mostly one genera, one genus, and that's the Mycobacterium. Okay, and these organisms actually have been quite successful. That's why we have a whole staining procedure just to be able to see them under the microscope. If you've heard of tuberculosis, that infection is caused by Mycobacterium tuberculosis. So clinically, some very interesting organisms in that genus. So it's important to kind of take a look at that. So what's special about these cell walls? And again, it's much more complicated than what we're going to talk about here. But the one really unique thing that they have them in their cell walls, in addition to peptidoglycan, is that they contain mycolic acid. Okay, this is a long chain fatty acid. Okay, so when you think about kind of the interesting properties that that's going to give the cell wall, actually makes it very hydrophobic. You could almost say gives the cell wall kind of a waxy, gives it kind of a waxy constituency or kind of a waxy feel to it. So anything, you know, you think about a sheet of wax paper, if you put a drop of water on wax paper, it doesn't soak into it, it beads right up. Okay, so that's kind of a good picture for these cell walls. Okay, so I want to come back to that in just a minute, This is actually a acid fast stain. You're gonna get it if you're live. You're gonna get a chance to do that next week. Because the staining dyes that we typically use to stain bacteria cells are dissolved in water, those water-based dyes don't adhere to mycobacterium very well. Again, waxy cell wall, lipid cell wall, high lipid cell wall. So we have special dyes, one that's dissolved in phenol that's going to penetrate those cell walls. We call these organisms acid fast because when they are decolorized, even with acid, they hold fast to the primary stain. So that's where that name comes from. And you know, I'll let your lab professor go into more detail there. But I wanted to pose a question. So I like this graphic because it kind of compares the three different kinds of cell walls here. So there's a few things here in this graphic that we didn't talk about. With the gram-positives, we didn't talk about that lipoprotein, but you see that, you know, enormous peptidoglycan there, just a little bit of peptidoglycan for the gram-negatives. And then the mycobacterium, right, there's some of this stuff that we didn't really talk about. There's our mycolic acid. Okay, again, a little bit of peptidoglycan. So I kind of like that schematic. So what do you think about for a second, and then we'll go back and talk through this. If you're thinking about, you know, organisms that would cause an infection, which one, or, you know, rank them in terms of how hard they're going to be, you know, how much they're going to withstand antibiotics. Okay, so which one of these organisms would be easiest to kill, which would be hardest? Okay, so I want you to think about that for a second. Take a minute, talk to the person next to you, see if you can, you know, defend what you're thinking. Then we'll see what you think. Knowing what you know. All right, let's see what you think. Anybody have any? What do you think? What's going to be the hardest kind to treat? Don't be afraid to be wrong. Agree. I think the gram-negative will be the hardest. Okay. What's your reasoning here? Okay, because of the lipid A and that causes inflammatory response. Okay, that actually is the second type. So we're exactly right, but you weren't exactly wrong. Okay, what do you think?
So we thought I said gram-positive just because partner said
okay, good. So what he said was his first inclination was gram-positive is hard because it has so much peptidoglycan. But then, well, maybe it's a permeability issue. Maybe that waxy cell wall. Okay, so good. I like what you're all thinking. Actually, yes, the mycobacterium is the toughest one to treat. If you know anything about tuberculosis, we only have a few drugs that works for that infection because we cannot get them to penetrate through that waxy cell wall of the mycobacterium. And kind of along the same lines with the gram-negatives, they have this other phospholipid membrane that's really kind of a little bit more difficult. We have less antibiotics that are able to work against gram-negatives. than what we do against gram-positive, just because of that permeability issue, getting the drugs in. Now, and then not to say that the gram-positives are easy, but remember this peptidoglycan, we liken that to a chain link fence. Okay, it's not selective as to what goes in or out. So it does, offer quite a bit of protection to the cell, but it's not choosy about what goes in and out. Now, those numbers may be different depending on, hey, if you had organisms that were sitting out here on the table, how long would each of them stay viable? That may be a different answer to that question. Membranes typically out in the environment are pretty fragile. They dry out really quickly. So that could be a different set of answers there. So good. So that's one thing that I kind of wanted to write here, but I wanted to wait a little bit. Makes it really impermeable to a lot of things, not just antibiotics, but disinfectants. We'll say more impermeable to antibiotics, disinfectants. It's tougher environmentally, actually, because it's helps it resist desiccation. Okay, so not as hard to kill as endospores, which we mentioned last time. We'll talk a little bit more about today, but typically a little bit tougher. Okay, more environmental stability. All right, so moving into the cell, past the cell wall. Not going to spend much time on cytoplasmic membranes. The membranes of bacteria are very similar to the membranes of eukaryotes. We have antibiotics that target bacterial cell membranes, but they're not much good because they're toxic to us as well. There's just not enough difference there between those of bacteria and those of us. So certainly a really important structure, though. It controls what goes in and out of the cell. It's that selectively permeable barrier. Maintains gradients for bacteria that don't have a lot of specialized organelles to carry out cell function. A lot of that happens in the cell membrane, or some of that, like their electron transport chains. Okay, for us, it happens in our mitochondria. For bacteria, all of those electron carriers and things are embedded in the membrane. Okay. So not a lot to say there about the structure. Of course, in terms of transport, we'll spend a little time talking about that. When a cell obviously has to bring nutrients and other things into the cell. And if that transport is passive, that means there's no ATP required. Okay, the cell doesn't have to spend any energy to get certain things into the cell. So some things can just easily diffuse. That means if there's something of a higher concentration outside of the cell, sometimes that can just diffuse right into the cell, move from greater concentration to lesser concentration. What kind of things can do that? There's some gases, sometimes simple sugars can just diffuse in, water. Anything that's soluble in that phospholipid bilayer sometimes can diffuse in, like alcohols. Oftentimes, though, there needs to be a specific protein channel for things to transport in. So if that's the case, we would consider transport facilitated diffusion. In other words, it's facilitated by some protein. That can be a very specific channel, or it can be the channel that, like, just lots of things, non-specific can come right through. And of course, osmosis, that's the movement of water, diffusion of water from a high concentration to a low concentration. So sometimes that might happen through a channel, but oftentimes that could go right through the membrane. So we need to review osmosis. It's a concept that we're going to come back to several times. So if this is something that you don't kind of have in your long-term memory, we want to find a way to get it there. I find it's a concept that students are still confused on, even though you've had this in other courses. I think part of the trouble lies with these terms, okay? Isotonic, hypertonic, hypotonic. If we're going to use those terms correctly, we use them to describe the solution that a cell is in. Okay, oftentimes if we've got movement of water, then it's going to be, the cell's going to be in some moist solution. So obviously isotonic, that means the solutes. So a solute is something that doesn't dissolve in the water and can't pass through the semi-permeable cell membrane. So oftentimes we talk about like salt or sugar as being solutes that can't pass. If the cell is in a hypertonic solution, that means outside the solution has a high concentration of some solute. Okay, so when we put brackets around something, that means concentration. Okay, so a high concentration of solute that's going to be outside the cell. So the question is, which way does the water move? A couple different ways you can think about this. One student said to me once, well, I remember it because water follows stuff. Okay, so if there's more stuff, more solutes outside of the cell, then water is going to follow that. Okay, and you get water leaving the cell. Okay, the cell is going to dehydrate in a hypertonic solution. Okay, how much it dehydrates depends on how big the concentration gradient is. But that's typically a really bad situation for any kind of cell. Okay, they need water for metabolism. So typically, again, they don't like those kinds of conditions. That's why we add sugar or salt to our food when we're trying to keep it for long periods of time, because that reduces the chance that bacteria are going to be able to grow there. Okay, so whether you got a cell like a bacteria or any kind of cell with a cell wall, or you got a cell without a cell wall, okay, they're going to be dehydrating. Another way you could think about this is if the solute concentration is high, then the water concentration is going to be low, and you know water moves from high concentration to low concentration. Okay, so it's higher in the cell, and so it's going to be leaving the cell. Hypotonic solution, right? That means the solute concentration outside of the cell is going to be lower. So that means the water concentration is going to be higher. So water this time is going to be moving into the cell. Okay, we'll say cell swells up with water. So now we see a difference whether a cell has a cell wall or not. Most bacteria have a cell wall. They are quite happy in a hypotonic solution. We call that a little bit of turgor pressure, a little net movement of water into the cell. That's fine for a bacteria cell. If you got something like a red blood cell, though, you get a little bit of difference there, and it's likely to lyse. Okay, so that's why you wouldn't give a patient an IV of just pure water, right? That's not going to be good for them. You always put a little saline in there to make it more isotonic. All right. So if that's confusing to you, sit down with me sometime and we'll talk through that again, because that's just kind of one of those concepts that you need to know. If a cell transports things using ATP, then we call that active transport. Okay, so ATP, spending of ATP is necessary. Okay, the energy currency of the cell. And I'm not too worried about whether we call this a uniport or anaport or a coupled transport. Basically, sometimes it's more energetically favorable maybe to let one thing diffuse out of the cell or pump one ion out of the cell, for example, so that something else can be brought in, again, with spending a little bit less energy. So that's kind of the difference between these three things. Again, I'm not too worried about naming what's what. Just active transport, ATP is necessary. And then there is one kind of active transport that is unique to prokaryotes. Okay. And this is kind of an ingenious system to make sure that the best example that I have of this is sometimes when glucose is brought into the cell. And you may recognize this when glucose gets changed to glucose 6-phosphate. That's the first step of glycolysis. Okay, so this kind of transport, it's called group translocation. It just means that the substance is chemically modified as it's transported into the cell. Okay, so like I said, glucose becomes glucose 6-phosphate. Why would it be an advantage to the cell to do that? Well, there's probably a lot more glucose inside of the cell than out. So it's transporting it against the concentration gradient. So when it comes into the cell, we don't want that glucose to just diffuse right back out again. So if the bacteria chemically modifies it, in this case, just by adding a phosphate, then that keeps it from diffusing right back out again. All right, so again, only prokaryotes do that apparently. All right, so moving further into the cell, cytoplasm. Not a lot to say there. You know, that's the stuff inside of the cell. If you're going to use these two terms correctly, cytoplasm and cytosol, I think a lot of times those terms are used kind of synonymously, like they're the same thing. But if you're going to use them correctly, technically the cytosol is the fluid present in the cytoplasm. So what else is in there? It's going to contain some proteins and ions kind of floating around in there, as well as lots and lots of fluid, lots of water. Okay. In the nucleoid region of the cytoplasm, remember we said prokaryotes don't have a membrane-bound nucleus, but they typically do have their DNA kind of all in one area of the cell. So we call that the nucleoid region. That's, again, where the DNA is located. Okay. It's not membrane bound. That's going to contain a chromosome. Bacteria typically will have 1 or 2 chromosomes, most often 1, sometimes 2, and that's going to be in a circular form. Okay, so we're going to talk more about that when we talk about our genetics, get into our genetics unit. But that's going to be much more favorable for the DNA. It's going to be much more stable. It's in a circular form and it doesn't have its ends exposed like a linear chromosome would. And bacteria also have DNA that's structured as plasmids. Okay, they're going to have typically a lot of those. Those are just smaller circular pieces of DNA. And usually kind of information is stored on plasmids, which kind is on chromosomes, usually anything that's necessary for the cell to metabolize or reproduce. That information is going to be on a chromosome. Anything that's just kind of extra that may give the cell a survival advantage might be stored on a plasmid. Okay, but we'll talk more about that later on. Not a lot else inside of the cell, but all bacteria cells do have ribosomes. Okay, so that's not something that's membrane bound. So all bacteria, all archaea, all eukarya, they have to have ribosomes. And of course, that's very important because that's where proteins are synthesized within the cell. Okay, what are ribosomes made of? They're made of ribosomal RNA. and proteins. And they are organized into two subunits. Okay, so you have a small subunit and a large subunit. Those come together when the messenger RNA docks on the small subunit. The large subunit joins that, and that's when protein synthesis begins. Okay, so you can see here, I thought it would be easiest just to compare the prokaryotic ribosomes with the eukaryotic ones. A lot of similarities there, but enough differences in the prokaryotic ribosomes and the eukaryotic ones that we have antibiotics that actually will only adhere to the prokaryotic ribosomes. So enough differences in structure there. So you can see here's a large subunit prokaryotes, the 5S strand and the 23S strand. Those are strands of ribosomal RNA. And then together with proteins, those add up to a 50S. What is that S? That stands for Svedberg's. I think that's an E. That is a unit of sedimentation rate. Okay, so what does that mean, sedimentation rate? Basically, we would put those ribosomes in a centrifuge tube, spin them down in a centrifuge. How quickly would they sediment down to the bottom? So it's not really, it's a measure not just of size, it's kind of a measure of size, weight, as well as shape. So it's not linear. You can't necessarily kind of add up sedimentation rates. So that's why when we have a large subunit that's 50S and a small subunit that's 30S, those come together to make a 70S ribosome. Okay. So yeah, it doesn't, I mean, it's kind of confusing. It doesn't add up, but it's just because it's sedimentation rates. And then the eukaryotic structure, again, is very similar, kind of different sizes of pieces of ribosomal RNA, but they are going to have the 80S subunit when that comes together. Okay. And archaea have ribosomes that are again more similar to the prokaryotes, to the bacteria. So we just consider them prokaryote, but there are some kind of structural differences that we won't get into there. Okay. Some bacteria, like I just alluded to, are able to produce this structure called an endospore. Okay, we mentioned this last lecture just as a way to classify some organisms because it's really not very common in the bacterial world for a bacteria to be able to make an endospore. There's really only two very famous genera that are able to do it, the Clostridium and the Bacillus. Remember what an endospore is. This is really a survival mechanism. It's not reproductive. So when we talk about the fungi a little bit later, it's a different story. When fungi make spores, they're making lots and lots of them. It's a reproductive strategy. For bacteria, they start out with one cell and they end up with one endospore. So not reproductive. When do these organisms do this? Just a little abbreviated cycle here of a spore forming organism. When the organism is metabolically active, we say that that's a vegetative cell. So metabolically active, that just means it's, you know, making energy and making cell structures, dividing. These organisms, the Clostridium and the Bacillus, sometimes they might go through this sporulation cycle just as part of their kind of normal lifespan. But we know we can encourage them to sporulate by having them encounter some kind of harsh environment. So if they encounter some harsh environment, some examples of harsh environments, things like high or low temperature. So extreme temperature, extreme pH, low nutrients. You can probably think of other ones, dry conditions, radiation, exposure to radiation. So any of these would be considered harsh environments and could start this process of sporulation where the DNA is replicated and then the spore coat develops around the DNA. The spore coat is made of lots and lots of layers of proteins, tough structural proteins like keratin, the protein that's in our fingernails, many different layers. There's 30 or 40 some unique genes that have to be present for the cell to be able to do this. Again, so it's not common, but eventually you end up with this free endospore that can persist for decades, hundreds of years, maybe even thousands of years. And when the conditions are right again, it would still be able to germinate back into a metabolically active cell. Okay, so it's a survival technique when the conditions are poor. Keep in mind, the process of sporulation takes between 6 to 10 hours. So you're not gonna hold a cell over a flame and bam, it's a spore and it's gonna survive that. Okay, it takes some time for that process to happen. Okay, but there's a lot of cool infections that are caused by spore formers. Bacillus, anthrax is a bacillus. Clostridia, things like tetanus and C. diff and botulism. There's a lot of really kind of cool infections. Most of them are harmless, especially the bacillus. Those are just soil organisms. Okay. So again, that's something that may be in the cell, probably usually not. You will get a chance to do a spore stain if you're in lab that will look something like this, where we get the spores to take up one dye. Those are small little green ovals are the spores, and then the metabolically active vegetative cells are the red rod-shaped organisms there. And then this is just what anthrax looks like. Usually we do a special spore stain because when we're just kind of doing, just throwing like one dye on those cells, like it looks like was done in that picture, the spores don't take up the dye very well. You can kind of see a few here and here, but they just don't stain very well unless you use a special staining procedure to do that. Okay, and lastly, in terms of prokaryotic structures, there's a few other things that are mentioned in the book that I'm going to skip over, like the cytoskeleton. Some bacteria have the ability to form inclusions. And usually these are to store some kind of, chemical or nutrient that could eventually be in short supply. So some things like lipids or starch are typically stored in inclusions. And one that I wanted to, that's particularly interesting, some bacteria, if you grow them in certain conditions, they will store polyhydroxybutyrate. All right, we can abbreviate that PHB. That's just we'll call it a starchy polymer. And so here's a picture here where we've gotten this, I think it's a bacillus organism, actually grown it in the right conditions so that like most of the cell is taken up by this PHB, this starchy polymer. And the interesting thing is we can use PHB to make plastics, right? So normally we make plastics, it's petroleum based. You throw that plastic in the landfill and it's there basically forever. So I just love thinking about a world where we could use bacteria to do things for us and make our life better. They found a way to make plastics out of this, get bacteria to make plastics for us. So again, I'll come back to the slide in a minute, but the big advantage here is, here's a plastic that's, thrown in the ground, and they, over a matter of weeks, I'm not actually sure what the timeline is. That was just my memory of when I read about this. That plastic will degrade, like down to nothing, because it's organic. So, not ideal for all applications. Sometimes that would be a bad thing to have whatever in the bottle, have your bottle disintegrate. But a lot of applications for that, maybe some medical applications, things like plastic sutures that would eventually just dissolve instead of having to be taken out. Water bottles. We don't need water bottles to persist forever. So that's kind of a cool thing. Unfortunately, it's more expensive to make plastics that way. And so in the near future anyway, that's probably not going to catch on. Another example of an inclusion, some bacteria will store gas in gas vesicles. So if this was an organism that was photosynthetic and needed to align itself kind of in the water column in the just right place, it could kind of adjust that by storing more or less gas in these vesicles. Okay. All right, so that brings us into eukaryotic structures. So we're still within chapter 3. I'm going to run through these a little bit more quickly because it's just sort of less our focus. And I realized I need to find a picture for this slide. This is kind of boring. But we talked about how bacteria will often, very commonly, secrete some kind of glycocalyx outside of their cell. Again, it's very common because the functions just make it really helpful, help the organism survive. Eukaryotic, some eukaryotes will do this as well. Apparently, only eukaryotes that don't have a cell wall. Okay, so cells without a cell wall will sometimes make a glycocalyx. So let's see, let me write that again. So only cells without a cell wall. If it's some kind of cell, like a plant cell or a fungus that has a cell wall, it's just not really necessary for them to make a slimy substance on the outside of that as well. Okay, so this means it's going to be anchored to the cell membrane. And it's usually a little bit more loosely defined than what the capsule would be of a bacterial cell, but some of the same functions as bacteria. Remember, for bacteria, main one was attachment. For bacterial cells to stick to other cells, as well as maybe some structure like our teeth. Same kind of thing for eukaryotes, attachment, some protection against drying out, just like for bacteria. And it doesn't really, eukaryotic cells are much less likely to be phagocytized. So that was the other function for bacteria. For eukaryotes, it fulfills more of the role of strengthening their cell surface. Okay, so they don't have a cell wall, so if they've got maybe a glycocalyx, it makes them a little bit more likely to withstand, changes in osmotic pressure, for instance, a little bit less likely to burst if they're in a hypotonic medium. Okay. And again, it's just some eukaryotes. Apparently, cells in our digestive tract have glycocalyces. So there's one example. All right, cell wall. We're not going to spend as much time here. But if you think, we haven't really surveyed our eukaryotes yet. That's our next chapter. If you think about what kind of eukaryotes have a cell wall, any guesses? I just mentioned one a minute ago. So plants have a cell wall. Whoops, plants, algae, fungus. Okay, those are the main ones. So All the time people get confused, right? The difference between plants and animals, plants have a cell wall, animals don't. Okay, that's different than the difference between prokaryotes and eukaryotes. That's all about, remember, having a nuclear membrane-bound nucleus and membrane-bound organelles. Okay, so there are eukaryotes that have cell walls. In terms of the composition, very different for plants. Very often they're going to be made of cellulose, fungus, another polysaccharide, chitin. Although there's a lot of variation there, sometimes there's other kinds of polysaccharides. Algae, I'm just going to say lots of variation. There's some algae that have silica in their cell walls. Okay, those are diatoms. So we'll look at those in a minute. There's a lot of variation. Do protozoa have cell walls? No. No cell walls. Okay, they will often have we'll call it a proteinaceous helical. Okay, they often have a helical which is made of protein. So they kind of have something around their surface, but it's not a cell wall. And remember, like I said, there's a lot of variation, but remember, never is going to have peptidoglycan. So, might have something that's kind of similar, but it's not the same thing. Okay. Same kind of function as what we saw in prokaryotes, strength, support, shape. Okay. And moving into the cell membrane. All right. There are differences in membranes structure. Animals, for instance, will have sterols for added support. Okay, in other words, steroid lipids like cholesterol. We kind of tend to think about cholesterol as being a bad thing, but it's absolutely necessary in our membranes. It helps them stay fluid when it's cold, or when it's warm, it helps them solidify. So that is one difference that you don't see in prokaryotes. Same kind of functions though, semi-permeable barrier. One difference here for eukaryotes, some eukaryotes can kind of reorganize their cell membrane and actually use it for movement. Okay, this is only going to be eukaryotes without cell walls, right? If you've got a cell wall around that membrane, then it's not, the membrane's not going to be able to move through that cell wall, obviously. Okay, so if it can reorganize into pseudopods, you think things like amoebas. Okay, that's how they move in amoeboid action. They're just reorganizing their cell membrane. And also in terms of transport, I kind of meant to make a heading right there. Okay, so movement, that's really protozoa that maybe could use it to move. In terms of transport, there are a couple of different things that bacteria cannot do. So let's just say this is unique to eukaryotes without a cell wall. Okay, bacteria almost always have a cell wall, so they're not doing endocytosis. This is transporting things by kind of extending pseudopods and engulfing that thing. Okay, so phagocytosis is when solids are engulfed. And a term that you hear less, pinocytosis, is when liquids are engulfed. Okay, and then obviously exocytosis is just going to be the opposite of that. Usually, you know, exporting substances, usually exporting wastes. Okay, so I don't think I posted this, or maybe I did, I forget. But here's an example of an amoeba extending a pseudopod in order to phagocytize some kind of other protozoa here, like a paramecium. Okay, so again, phagocytosis is a kind of endocytosis. Obviously, that amoeba does not have a cell wall, otherwise it wouldn't be able to extend those pseudopods. Okay, so bacteria, if they want to eat some big object, they can't do that. They have to secrete enzymes to break it down and then be able to transport it in smaller pieces. All right, next structure we would come to as we're making our way in then is flagella. All right, so again, some eukaryotes can have a flagella. For eukaryotes, we consider it an internal structure because it's actually on the inside of the membrane. Okay, membrane bound. I think I alluded to this when we were talking about prokaryotic flagella. Eukaryotes that have a flagella, this is just mostly protozoa, it has the same function as prokaryotic flagella, meaning for movement, but almost everything else is different. The entire structure of it is different. Just about everything else you can think of is different. So the first thing is it is bound in a membrane. It's composed of microtubules. So if you remember the prokaryotic flagella, I'll just put that in parentheses, prokaryote, that was made of many different units of flagellin. I think that I wrote that earlier, but just in case I didn't, I wanted to write it again. So prokaryotes made of flagellin, eukaryotes, microtubules, which is made of the protein tubulin. All right, so different protein, and we describe these microtubules as being in a 9+2 arrangement. And that seems like kind of a weird way to write that. I remember having a conversation with my son when he was younger, and he said, why don't they just call it 11? Okay, math genius, I know. And I said, well, there's a good reason they call it a 9+2 arrangement, because there's actually 9 pairs of microtubules around the outside and then 2 single ones in the center. Okay, so they call that a 9+2 arrangement of microtubules. And that's in the filament portion. There's really no hook. If you remember, the bacteria had kind of a hook region there as it transitioned into the basal body. We'll say here in the basal body, the flagella are considered a 9+0 arrangement. Okay, so for some reason that arrangement changes a little bit. How that does exactly is probably very complicated, but down here in the basal body, the part that's kind of really anchored into the cell, there's triplets, 9 triplets and none in the center. So that's why they call it 9+0. Okay, so a little different structure. The movement is quite different as well. For eukaryotes, the flagella move in a whip-like motion. And if you remember, the prokaryotes spun around, okay? So clockwise is one direction, or counterclockwise is one direction, clockwise is another. Okay, so it's kind of hard to see that motion in these videos because they move so fast. Let's see if I can bring these up successfully. This particular animation is like 11 minutes long. So, you know, you can watch the rest of this later if you are so inclined. But this is kind of, let's see. I forgot to write down where it was most interesting. Sorry. Well, you can kind of see here. There was this really cool part where the flagella was sticking out, and if you're interested in seeing flagella movement, you can watch this. Usually eukaryotes are only going to have like 1 or 2 flagella. They're not going to have flagella all around their body. This one in particular compared, here's an example of cells in our body that have flagella that you may have forgotten about.
Here you see some bacteria swimming around. You'll notice the flagella go 360. They're like little outboard props. See, it's going around and around and around.
Okay. So
here we've got a human sperm, and there's a flagellum coming out of the back. And you'll notice that it goes back and forth in a wave motion, as opposed to with the bacteria, where you had it turning 360 like a little propeller prop. So that is the difference between the motion of bacterial flagella and eukaryotic.
You can play around that with a little bit. There was.
Here you see some bacteria swimming around. You'll notice the flagella go 360. They're like little outboard props. See, it's going around and around and around. Here we've got a human sperm, and there's a flagellum coming out of the back. And you'll notice that it goes back and forth in a wave motion, as opposed to with the bacteria, where you had it turning 360 like a little propeller prop. So that is the difference between the motion of bacterial flagella and eukaryotic flagella.
It can go both ways, but okay. So anyway, moving on. A related structure that we see in some eukaryotes is the cilia, and these are much shorter than flagella. They are still functioning to either move the cell in the case of protozoa, or there are some eukaryotic cells that have cilia, and they would have them to circulate materials. So we have ciliated cells lining our respiratory tract, and those cilia, the function of them is to propel things, up and out of our respiratory system, pathogens or dust or whatever. If a cell has cilia, there's typically a lot of them, somewhere between 100 and 1,000 on the surface. And again, this is going to be mostly, we think about protozoa having them, a few types of animal cells. And I'll also say that the structure is similar to eukaryotic flagella. And the movement certainly is the same as eukaryotic flagella. So the cilia are going to move in a whip-like fashion. Think about it. If they spun around in circles, think about how would that cell, move in a purposeful way if all those cilia spun. What good would it do if our cilia and our respiratory tract moved in circles? That wouldn't make much sense. So that's what helps me remember that eukaryotic flagella and cilia move whip-like fashion, prokaryotes spin. All right, this list of organelles, I'm just going to run through these really quickly. Probably, I really just want you to have a basic idea of what the organelles do. Take a good cell biology class if you want to go into more depth here. Obviously, the nucleus contains DNA, and you can read more about these if you want in your textbook. Again, just basic function here. The ER, kind of a transport system. The rough ER has ribosomes. So proteins that are made on the rough ER typically are ones that are transported outside of the cell. They follow through that transport system of the ER and into the Golgi and often out of the cell. The smooth ER, lots of different varied functions there, some synthesis reactions going on, no ribosomes there. So that's, we'll keep it simple. Golgi, we're going to come to that next after something's passing through the ER, kind of a additional modifications. Maybe it's adding a lipid onto a protein to make a lipoprotein, or a sugar onto a protein to modify it. And then two organelles that we'll talk more about in a minute. The mitochondria, obviously that's the powerhouse of the cell where we make ATP. And in photosynthetic organisms, they have all their pigments concentrated in the chloroplast, where they're harvesting light. Another organelle that I want to spend just a brief more time on is the lysosomes, because we'll talk more about these later on as we think about our white blood cells playing an important role in our innate immunity. But these are really important. If a cell is ingesting some bacterium, say our white blood cell, and that bacterium is going to be enclosed in some membrane-bound vesicle, how is that bacterium killed? Well, our lysosomes here are going to fuse with that phagosome. And within the lysosomes are all sorts of chemicals that are made and digestive enzymes that are going to act to kill that invading bacterium. Okay, so contains digestive enzymes. And then lastly, I want to talk about the endosymbiotic theory as the end of our chapter 3 material. This is a theory that attempted to explain how eukaryotic cells, cells were made, or first how eukaryotic cells came to be. Okay, we think the very earliest cells were prokaryotes. So how did they make the leap to become more, complicated cells and thus more complicated organisms? Well, this theory was put forward by Lynn Margolis in the 1970s. And when she first presented this at conferences, she was just laughed out of the conference hall. People thought it was ridiculous, but over time, we learned more and more, and the evidence started to pile up, and now this is generally accepted. But so the theory says that an early anaerobic prokaryotic cell ingested an early prokaryotic cell that had the ability to respire aerobically. An endosymbiont is an organism that lives inside another organism. So instead of just, digesting this organism for nutrients, this second cell actually acquired the ability to live inside of that cell, and that gave rise to the first eukaryote. I keep wanting to spell that wrong. Okay. So what other sorts of things are going to support this theory? Well, we think this endosymbiont now has become either mitochondria or chloroplasts in cells. So what supports that? Mitochondria and chloroplasts, they actually both have their own DNA. And it's in the form that's common for prokaryotic cells. Okay, it's circular. Mitochondria and chloroplasts both have their own ribosomes. And it's in the form of that we see in prokaryotic cells. Okay, they have 70 S ribosomes. Okay, so that's really kind of very interesting. they both replicate independently of the cell. Okay, and they are about the same size as a typical prokaryote. Okay, so all those things really kind of point to, hey, maybe those organelles, mitochondria and chloroplasts, used to be bacteria. And now they have kind of evolved to kind of live with a cell. Now, not everything, suggests that. A lot of the DNA that are necessary to replicate those organelles has been transferred to the nucleus, and it's not in those organelles, but there's a lot of evidence to support that. So, and that's known as endosymbiotic group. Okay, so that ends chapter 3. I thought I was going to have a little more time than that. But that's as much as I'm going to cover through the exam on Wednesday, and your homework chapter 3 is through Wednesday. I think probably, I know we have like 4 minutes left, but I think probably that I will stop there, and I will, after the exam on Wednesday, I'll start chapter 12. Okay, so that's talking about different eukaryotes in chapter 12. All right, so I'll be up in my office for a while if you have any questions. Otherwise, have a good day.